Hello students, welcome to the channel Basics of Biology. Today we are going to start a discussion about chapter 5, the fundamental unit of life. Today's topic is microscope. So let's start. There are many types of microscope like simple microscope, compound microscope, electron microscope. Okay. But the two types of microscope that are commonly used are compound microscope and electron microscope. And you just need to know about compound microscope, which is used in the lab. First one is the eyepiece. The eyepiece is the part of microscope that magnifies the image produced by the microscope's objective so that it can be seen by the human eye. Let me explain. This is the eyepiece and it magnifies the primary image which is produced by the objective. If you put your eyes on this eyepiece, you will see the large image of small specimen which you want to see. And this eyepiece can be monocular or binocular. The monocular has only one eyepiece, whereas binocular have two eyepieces. Okay, let me show you. So students, these are two types of microscope. Here you can see in the first image there are two lenses, and the second image you can see there are one lens. Means first one is the binocular and the second one is the monocular. Okay. And the next one is the arm or carrying handle of a microscope. So students, this is the arm of a microscope. It supports the microscope head and attaches it to the base. And one more feature of this arm is that by holding it, you can move microscope anywhere. And the next one is a base of microscope. So students, this is the base of a microscope. A base of the microscope that houses the illuminations and supports the microscope. Next one is nosepiece. It's also known as the revolving turret. It holds the objective lenses and attaches them to the microscope head. So students, you can see this black portion. This is the nosepiece. Next, objective lenses. So student, these are objective lens. There are usually 3 to 5 objective lenses on a compound microscope, each with different magnifications level. This objective lens helps to zoom the image of specimen. And in a compound microscope, commonly used magnifying powers are 4x, 10x, 40x and 100x. Okay. Next, specimen and slide. I have already told you about the slide or other equipments used in the lab. You should watch the video. I will provide the link of this video in the description box. Okay. Let's see what is a specimen. A specimen is the biological sample or organism that used for scientific examination and study. Next one is a stage or platform. So, this is the stage of a microscope. The stage or platform is that part of a microscope upon which specimen or slide are placed. Okay. Next one is stage clip or slide holder. This is the stage clip or slide holder. What is the function of this clip? It holds the slide on stage. Next one is aperture disk or iris diaphragm. This is the aperture disk. It is the hole in the middle of the stage through which light from the illuminator passes to illuminate the specimen. Okay. Next one is illumination. It is the light source of a microscope. And the microscope you can see it is used a bulb as a light source. But earlier microscopes used mirrors that reflected external light into the base of the microscope instead of halogen bulbs as their source of illuminations. Next, coarse adjustment and fine adjustment. Basically, coarse adjustment helps to move the stage up and down and bring it closer to the lens. And by using fine adjustment knob, you can make small adjustment as it bring the object into focus. Here you can see the knob. Next one is condenser. Students, this is a condenser. Okay, this lens condenses the light from the base illuminations and focuses it onto the stage. This piece of the compound microscope sits below the stage and typically acts as the structural support that connects the stage to arm or frame of the microscope. Next one is bottom lens or field diaphragm. So students, this is a knob which is used to adjust the amount of light that reaches the specimen or slide from the base illuminations. Hope this video will help you. Thank you for watching.